Colorimetric method of blood glucose estimation. First of all, I start with arranging my materials. As you can see, that is the color reagent 1, and my glucose standard, the blood sample. The tubes you are seeing here on this rack was labeled test and standard, and I have my blank tube. That is the test tube 2 and test tube 1. As you can see, there are 2 patient sample which is in the fluoride oxalate bottles. So the sample has been well spun. As you can see, the plasma is separated from the packed cell. So I'll pick 10 microliter of the patient sample, which is the plasma, pipette 10 microliter into the tube labeled test, and that is for the first sample T1, and I'll also do same to the second sample, which is the T2. The same microliter of blood, 10 microliter of the plasma. Thereafter, after picking the sample into the test, except for the second sample, I'll go in with the next reagent, which is the standard, and that is 10 microliter of the standard reagent. We put it into the standard tube. That is the standard tube as I showed you earlier. After that, I ensure I dispose my pipette tips and cover my reagents properly. After that, I'll go in with my colored reagent, which is the R1. I'll ensure I pick 1000 microliter of the colored reagent into the 3 labeled tubes. As you can see, that is the colored reagent R1. So I'll pick 1000 microliter into each of those tubes. After adding the color reagents, as you can see, in the right proportion into the tubes, the 3 tubes, as you can see, I ensure I mix the samples thoroughly, after which I incubate the sample at room temperature for 15 minutes. That is me setting my timer, and I look at the samples for the color change while mixing it consecutively. Then I go to my spectrophotometer. That is a spectrophotometer. I connect it to the light source. Then I turn it on and I ensure I zero my machine with my blank solution. As you can see, I set my wavelength to 500 nanometer because that is the proper wavelength for this particular test. That is me trying to zero my spectrophotometer, that is to balance it. Then after, as you can see, it is balanced. The figures there are 0, showing that it is zeroed. So after that, I will make sure I check on my samples. Then I bring, as you can see, the color change. Then I bring them to my spectrophotometer. Then I go in with my standard. That is me pouring the sample into the cuvette, and I read my standard result, absorbance of the standard. Then I record it. After that, I go in with the test. That is for the first sample. That is me trying to dab my cuvette, and I go in with the test, the sample. I put it into my spectrophotometer. Then I read the absorbance of the test. One thing I want you to know about colorimetric method of glucose estimation, it looks cumbersome. Yes, it is. And that is why most labs make use of the glucometer, which is a machine that all these reagents is being impregnated into it, and it becomes a kind of rapid diagnostic test. We'll go over to calculate the outcome of the test. Thank you for watching. Do well to like, follow and share my page.